Okay, now let's get started. And before we dive into the application cases in different fields, let's first take a look at the technical background of the DNA methylation. For the decades, DNA has been seen as the key material that determines the genetic information in living beings. But the new studies in recent years show that the genetic information is not fully decided by genes. For example, researchers have been found that we can change the modifications of genome without affecting the DNA sequence. And these changes can not only influence an individual's development, but also can pass on the next generation. So just like these two biological phenomena shown here, the twins have the same genotype, but their personalities and the habits and the other phenotypic informations are often different. And another case is about the bees. And the bees are developed from the same larvae, and they have the different outcomes due to the different foods. And in the 1942, the compact of the epigenetics was first put forward to define the phenomena of the phenotype change, but without the genotype change. A wide variety of the epigenetic modifications have been discovered so far, and they have different modification situations at different molecular levels, such as the DNA levels and the RNA levels, and also on the histone and the chromosome levels. So among the many types of the epigenetic modifications, DNA methylation is one of the most started. Studies have shown that there are range about 5 to 1,000 base pair in the mammalian genome that are rich in the CpG, and about more than half of the coding genes contain the structure. Methylation modifications in the CpG of some gene regulatory elements, such as the promoter elements, can prevent the transcription factor binding to DNA, thereby to affecting the gene expression. So the studies have shown that the DNA methylation can play many roles. It can participate in the maintaining the stability of genetic material, and it also can participate in the regulating gene expression, and also can regulating the X chromosome interactivation, and also can participate in the formation of the alleles genes. It is also related to the embryonic development and the cell totality, and also occurrence of the disease and many other life activities. And the DNA methylation plays an important regulatory role in life, and it is considered as the fifth space. So the methylation-related research is also gradually increasing, as shown here, and there are many different detection technologies for DNA methylation, such as the methylation macroarray and the targeted bisulfite sequencing, and also about the whole genome bisulfite sequencing, which can be called as namely as the WGBS. So the WGBS, comparing with the other technology shows here, is used to detect the methylated cytosine by treating the DNA with some like the bisulfite treatment before we do the sequence. The WGBS has become the gold standard for starting on genome-wide methylation at the single base resolution. So most studies have used WGBS technology to detect DNA methylation and provide the effective data support for various studies. The main research directions for WGBS including like the cancer research and the growth and the development and the environmental impact, and also about the reproductive genetics. So in the next part, we will see some research cases in these different fields. And the first topic I want to I want to talk is about the tumor early screening biomarker. And in the cancer, the high methylation of CpG isolate in the promoters occurs about 10 times more frequently than the gene expression loss caused by mutations. But about 16 to 18% of CpG sites are methylated in the normal cells. But in the tumor cells, the overall methylation level drops to about 20 to 15%. So compared to the normal cells, the methylation state of about 10 to 60% of the CpG size may have changed. So there is a type of DNA called cfDNA in the body, which means the circulating free DNA. The cfDNA refers to degraded DNA fragments that are released from human tissues into the circulatory system, such as the blood. And it is a new type of tumor molecular biomarker. The circulating tumor DNA, which means the ctDNA, refers to the cfDNA that comes from the tumor cells because it is stable and it is consistent, and the cfDNA methylation is an ideal way to screen for the cancer biomarkers. So in the early stage of the cancer development, when patients have not yet shown the clear clinical symptoms, but the changes in the state of the DNA within cells have already occurred. And these DNA fragments are released into the plasma, giving it an important information related to the cancer. So about the non-invasive liquid biopsy technique targeting the cfDNA have become one of the hotspots in the field of the tumor research. Because of its advantages, it has been widely used in different fields like the early screening and the early diagnosis and the tissue original of ctDNA and in the other kind of fields. And there are many different studies have found and confirmed the methylation biomarkers for many different types of cancer based on cfDNA. So like the slide shows here, this include about the breast cancer, like shows here in the pink box, and the lung cancer, like in the blue box, and also about the other kind of cancer. And here shows some the other types of methylation-related disease biomarkers. They cover the wide range of diseases. And also there have many commercial disease screening kits have also been developed based on these methylation signals. And next, let's take a look at some cases. And in this paper, the research team had developed a multimodal method called the sports meds, and it detects the tumor by looking at DNA methylation. It checks the methylation, the DNA copy number, and the end partings of cfDNA. In this way, the cancer can be found, and its locations and about the locations can be known from just one test. And the researchers also use the multi-feature model to make the test result more accurate. The sport mass model they developed combines the multimodal analysis of DNA's of the cfDNA features with a new machine learning method, and this help the it will give some helps with the early screening of the tumor. And in another study on the liver cancer, the authors collected the plasma sample from the Chinese patients and also from the health controls whom chosen at the rhythm. And they had a training cohort and the validation cohort. First, they compared the methylation levels of the CpG size in the liver cancer tissue and the normal blood DNA at the whole genome level, and they also picked out the most different sites. They tested the methylation of a small amount of ctDNA from nearly about 2,000 liver cancer and the health people's plasma. After figuring out the methylation levels of these sets, they did a lot of statistical analyses. They also used the different machine learning method based on AI. And in the end, they picked out about 10 and 8 sites to build a diagnostic model and a survival production model. The diagnostic model worked very well in both the training and the validation groups. The authors also shows that this model can tell us about the liver cancer apart from like the normal controls. In addition, the model can also accurately reflect the stage of the liver cancer patient and also can tell us whether there are any tumor residue in the bodies and the response to the treatment and whether there is a recurrence or not. So in addition to early screening for tumor, the methylation signals can also be used for the early screening and the diagnosis in the other disease. It is because the overall and the local methylation situation will be changed in the disease tissues. So in this study, the researchers analyze and test the methylation of cfDNA from the plasma of the pregnant women. About 1/3 of them have had the early onset PE. They had developed an early pregnancy risk production model, which correctly predict about like 72% of the early onset PE patients. The study collected 16 patients diagnosed with the PE and about 38 women who gave birth at a term as the control group. So after comparing and after the analyzing, they found that there is a significant difference in cfDNA between the PE patients and the control group. And the researchers also found that a large part of the DNA methylation changes in the PE patients were related to the placenta, and nearly in the early onset PE was accompanied by the delayed or the abnormal placental development. So after finding the difference in the cfDNA methylation between the PE patients and the control group at the time of the diagnosis, the researchers wondered if they could also screen for the disease early by looking at changes in DNA methylation before the symptoms appeared. So they found that the most PE-related DNA methylation state in the first 3 months of the pregnancy seems different from what was detected at the time of the PE diagnosis. So they further test and whether these changes in DNA methylation could be used to predict the occurrence of PE in the pregnant woman. So happily, the test results show that DNA methylation information around the 12 weeks could identify the risk of PE in the early pregnancy. And the researchers also further explored the mechanism and the regions for these changes in DNA methylation signals. And later, and at the last part, the researchers collected data for nearly about 200 pregnant women to validate the model. The conclusions were the same as what they found before. The score of DNA methylation signals decreased as the pregnancy age increased, and it was significantly related to the pregnancy age of the control group. And the researchers said that the model is a supplement to existing production models and also can prove the analyze of the DNA methylation signals can improve the scientific of the PE production. So in addition to playing an important role in the occurrence and the development of the disease, methylation is also present in the physiological process of the individual growth, individual development, and the aging. So at present, the mainstream theories about the aging can be divided into two major schools of thought. The first series believes that aging is a strictly regulated and programmed process. Its pathological consequence is an extinction of the normal physical process, such as the growth and the development. And the second theory believes that aging is the result of the lifelong accumulation for the damage and the rhythm areas, which eventually damage the tissue repairability. But no matter which series it is, some common cellular and the molecular defects in the tissue degeneration process related to aging are considered as the signs of aging. So some researchers have proposed about like 12 signals for the aging. They are like the genome instability, the epigenetic changes, and the other signals shown here. So among these many aging biomarkers, changes in DNA methylation in the epigenetic have become a hot topic in the field of aging research. The DNA methylation can change the structures of the chromatin and the conformation of the DNA, and also can change the stability of DNA and the way DNA interact with the proteins. Thereby, they can control the gene expression. So regarding aging and methylation, there is a review. The article summarizes the various aging mechanisms in which methylation is involved and deeply analyze the connection between aging and the DNA methylation. It also concludes that the aging process is accompanied by an increasing in the variance of DNA methylation. At the same time, article also can tell us the drug assistance and the increased exercise can effectively affect and delay the aging process. And in addition to being involved in disease occurrence and development and growth aging in the animals, the DNA methylation can also play an important role in the plant research. In the research and applications of plants, DNA methylation is mainly used in the plant's growth and development, as well as the adaptive phenotypes, such as the phenotypes under the environmental stress, like the jolt, the high temperature, and the low temperature, and so on. So in this part, we will share 3 studies in the plants research. The first one is also a review, which discussed the research process for the regulatory mechanism for the plant DNA methylation, and also about the biological significance brought by methylation research. The plants usually contains 5 functional DNA methyltransferases, which namely the MET1, CMT2, CMT3, and the DRM1 and DRM2. The methylation pathway caused by different methyltransferases are different. So in addition, the studies on various plants mentioned in the article have also revealed the novel and the deserved biological functions of DNA methylation. And the authors propose that since DNA methylation can be inherited and the phenotypic variation can be caused by epigenetic mutations, and also can caused by the epigenome editing, can create additional phenotypic variation on the same genetic material, and has a great application potential in the crop breeding fields. And in the terms of the plant growth and development part, although the mechanism of the 5 DNA methyltransferases mentioned before, we have been widely studied. but it is not clear whether they are sufficient to maintain DNA methylation throughout the genome. So to solve this problem, this article uses a completely new method. The authors successfully construct a plant model lacking DNA methylation. So by using the gene detecting technologies, the researchers can accurately target DNA methyltransferase genes, so allowing the plant to grow and develop without the DNA methylation. So this research material provides the scientists with an ideal model to detectly study the function of the DNA methylation in the plant gene expression and the development fields. And the last paper is about the environmental stress. So we all know the drought can cause the serious damage to the growth and the productivity of the plants. And also it is one of the most harmful environmental factors. The plants have involved complex regulatory network and the drought stress resistance. So the changes in DNA methylation can alter the gene expression and the chromosomal interactions in the plant's response to the environmental stress. So this review summary the latest research founding on the role of the copDNA methylation and the drought stress, including the methyltransferases and the demethylases, and also include the changes in overall methylation and the regulation of gene expression, and also about the RNA-dependent DNA methylation, like the AS event and the long non-coding RNAs. So that's all about the cases studies on the common research directions to the DNA methylation. And if we classify the research content according to the omics technology, for example, if we perform the joint analysis for DNA methylation and the transcriptomics, we will get an effect greater than the summary of their parts. So this is what we usually call the multi-omics research method. And here is an example for a multi-omics study using the epigenetic and the RNA-seq to explore the epigenetic features of expressed on the L1 local. So through the DNA methylation, we can find regions where the methylation levels had changed. It also can be called like the DMR, the different methylation regions. And we can identify the genes, the elements, and the various functional regions that are associated with the DMR in the terms of location. So such as the promoter, the enhancer regions, and so on. And the transcriptome can observe whether the expression of genes has increased or decreased. So by combining this result, we can obtain the genes that have changed at both of the methylation and the expression level. Further analysis for the functions of these genes can help us better understand the biological mechanism behind the phenotypic phenomena. So in addition to combining with the transcriptome, the DNA methylation can also be combined with more epigenetic techniques, such as this ChIP-seq and the AT-seq, and also can include the HiSeq to achieve the research growth. So for example, in these articles, the G9A is a methyltransferase that regulates the epigenetic modification, the transcription, and the genome organization. However, it is not clear whether these functions are dependent on each other or represent different functions of G9A. So in this study, the researchers used the above multi-omics technology and found that how G9A regulates the epigenome, the transcriptome, and the high-order chromatin structure through the different mechanism. Okay, so that's all about the case study for the different fields. And finally, let's move on to the summary part. And first, I want to do some summary about the research approach. Here you can see there is some information shows on the slide. And first, I think we can try to ask a question. What factors or genes that lead to change in the changes in the methylation, and then we can look at the changes in the methylation or demethylation enzymes genes, and then the next step we can find the genes involved in methylation level up or down, and in the following part we can confirm the specific reader proteins that reorganize the methylation changes signals. And next part, we can combine the transcriptome to find changes in the gene expression due to the methylation. And in the finally part, we can use the findings we found to try to explain the cellular changes, like the phenotypic changes and the changes in the clinical index. So here, as you can see here in the slides, we have achieved a closed loop for our research. And most methylation basic research will follow this tool, but not every step will appear. So in the most cases, it always a combination of the several steps. Okay, and the another summary part is about the data mining. And there is a logic for the methylation data mining part. It will generally follow these several steps. And the first step, we need to analyze the overall changes in the whole genome methylation, including changes in average methylation levels, the distribution of the methylation levels, and also can do some clustering analysis and the correlation analysis between the different samples. And the second part, we can do some analyze the differences in methylation levels to screen for this specific differential genes, including the DMR identification and the transcription of the DMRs on the genome alignments, like the TF banding analysis for the DMRs and the functional analysis of the DMGs. So the DMG means the differently methylated genes. Okay, and in the finally step, we can conduct the multi-omics integration, such as the methylation, the transcriptomics, or correlation analysis, including overall the association of the metagenes, the corresponding association of the DMG to the DEG, and the DEG, which means the different expressed genes. And also we can do some network association between the different omics. And okay, let's go move on to the family part. And there is the result validation part. In some, there will have some simple validation method, include the target gene DNA methylation sequence using the TBS technology, and we can also detect the MR expression level of the target genes using the qPCR technology. And we also can detecting the Western blot to make sure the protein expression. And there also will have some more complex validation method. Can include as the knockdown, knockout, and the overexpression of the DNA methyltransferases. And also we can do some DNA methylation inhibitors, and even do some experimental method based on no sequencing platforms, like shown in the slides here. And I think that's all about the webinar I want to share with you. And thank you very much for the listening and watching. I hope today's sharing can be helpful for your research.